This is TWIS. This Week in Science, episode number 858, recorded on Wednesday, January 12th, 2022. How to transplant a pig heart. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with dolphins, sharks, and ponies. But first, thanks to our amazon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. You, without meaning to offend anyone else who might be listening, you are a current modern human. But what is a current modern human? Mostly hairless ape descendant made up of a collection of odd ape descendants who over time added their genetic makeup to form that which you consider to be you. And what are you? Most philosophers would agree that you are a construct of consciousness, a self-aware, squishy brain creature living in a shell of bone, and reliant on a system of anatomical parts, all working together to supply you with a steady diet of human blood upon which to feed. Everything you know of the world beyond is through intermediaries, senses that report back information that is stored, correlated, interconnected, and analyzed within the neural network that is you. And when something goes wrong, when a part malfunctions, when a limb, nerve, or organ begin to fail, it can jeopardize the comfort, quality, safety, and security of squishy brain creature life. And so, you do the thing that squishy brain creatures are so adept at. You seek solutions. How best to keep the blood flowing, the senses reporting, the anatomical parts operating at optimal levels. At times, the solutions are simple. A new pair of glasses, a brace for the knee, medication for an ailment, a vaccine even to preempt a problem from happening in the first place. And in an extreme situation, you may even need to replace your heart. If you do, that new heart, that new part, that new blood pump, regardless of where it originated, becomes yours. Because you are more than a collection of parts. You are the very model of a modern squishy brain creature living in a shell of bone feasting on human blood. And your best tool for surviving the world is This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are here through the magic of modern technology. All of us collections of squishy brain folk. We are the model of a modern squishy brain. I really got to figure out how to make that work. One of my favorite musicals. Anyhow, thank you all for joining us for another episode of This Week in Science. And I am so excited to talk about all the science that we brought to the show tonight. So much of it out there in the world. I brought stories about spots on the moon, dark spots on the moon, zoo air, and fingerprints. See at those fingerprints. What do you have, Justin?
First human ever versus a volcano. My little medieval war ponies. How hemp can prevent COVID. Better government through drug-fueled drinking. And a new segment that I call My Problem with That.
Oh, I can't wait for that one. Maybe that's one we can all pick up as we move forward this year. My problem with that. Oh, I like it. Okay, Blair, what is in the animal corner?
Yes, I have dolphin anatomy, dogs wearing headphones, and spiders wearing hats or pants, depending on where they'd wear them. We'll talk about it.
Okay. Costuming for spiders. I like it. Well, we all like our little bit of costuming, and I also like our little bit of science here. And all of you, if you have not yet subscribed, make sure that you look for us on all the podcast players, This Week in Science or TWIS. We are also on Instagram and Twitch and Twitter as TWIS Science. We live stream from Twitch, Facebook, and YouTube. You can find us. And our website is twis.org. That's where you can find all the details that you might want to look for. But now it is time for the science. So let's dive into all these stories that we have teased in the last few moments. All right, we're just going to go right in to pig hearts. We're jumping in. We have talked about animal transplants. We've talked about pig hearts. We've talked about all this stuff before on the show. They did it finally.
Yeah.
Pig to human transplant. One of my favorite headlines, though, was from The Onion, where they said pig dies while waiting, or no, anyway, there was
For
a
a
funny
human body?
there was like a very funny headline from The Onion, which I highly recommend. But anyway, pig heart transplant. What did they do? How did they do it? Why did they do it finally? So turns out the gentleman who has received this xenotransplantation is named David Bennett. He's 57 years old, and he had advanced heart failure and was not eligible for a heart transplant because he hadn't done things to take care of or taken steps to take care of his body and his heart. Knowing that, the researchers petitioned the US FDA to authorize the pig heart transplant. Now, the heart came from a company that's called Revivicor, and Revivicor has been working in this area for years. They have been implanting pig hearts into baboons. They have been looking at all sorts of xenotransplantation possibilities. And for this particular pig heart, it has had multiple gene modifications. So they knocked out 3 genes that are enzymes that allow pigs to synthesize sugars that our bodies recognize as foreign. And so that gene modification basically gets rid of those sugars so that our body can't latch on to those markers and go, hey, we don't want you, reject. Additionally, there were human genes added, anti-inflammatory genes, genes for blood coagulation and prevention of blood vessel damage, and regulatory proteins related to immune and antibody response. And then finally, they removed growth hormone because they don't want the pig heart to continue growing inside of anybody's chest, because that can cause heart failure and other problems. So let's just stop it from growing. Yeah, it's already a big heart in there. Let's just calm yourself down. So the researchers, back in September, we talked about this, the possibility of having these modifications and how they could work in baboons. But now, yes, they have successfully transplanted this heart into this male human, 57-year-old male human. The interesting aspect of these hearts, too, that this heart had to be kept alive and kept happy in a bath of nutrients, and it had to be prepared and ready to be put into the human body. So it was they stored it in a cocktail that was commercialized by a company out of Sweden called Ex Vivo, and basically it gets bathed in a nutrient broth that has water, hormones, adrenaline, cortisol. And then the fun one, cocaine.
Good for the pig heart.
Good for the pig heart.
I guess.
Apparently, it keeps the pig heart fresh.
Okay.
So this
Is that true after it goes in also?
No, after it goes.
Okay.
I mean, I'm like, suddenly you get a pig heart and suddenly you have a need that you
Suddenly
didn't
it's
have
the 1980s again.
previously. Yeah. I feel like I'm living 40 years in the past. Yeah. But this is so now this gentleman has to be also on a rejection immunosuppressant. It's called KPL 404, and it'll shut down antibody production that basically these antibodies usually lead B cells to talk to T cells to get the immune response going. And this guy is going to be on this immunosuppressant regime for as long as he has this heart. And they're also working on this immunosuppressant KPL 404 as a treatment for rheumatoid arthritis. Lots of interesting aspects here for suppressing the immune system. But in this particular case, it's so that the body will want to keep the heart working and in the body, not rejected.
So the thing I find most intriguing is at some point, like our very early part of your telling of the story is, congratulations, Mr. Bennett. We've figured out your heart problem can be solved by giving you a donor heart. Oh, hey, that's great news. Yeah, this is going to work out fine. Yeah, the only thing is you have to, and I don't know the details for sure. I'm speculating. You have to quit smoking. Ah, okay. Yeah, then you'll live a long time. Okay. So what else you got? Oh, that's kind of it. No. Come on. There's got to be something else. Well, there is this thing where they're looking to put a pig heart in a human being for the first time, and it's never been tried before, and we don't know if it'll work. Yeah, that one. Yeah, that's the one. Do that one. I want that one. Failed to take care of himself for the head. That's the only thing I could think of. And
the conversation must have been an interesting one. I mean, this is going to give this gentleman an unknown amount of time. His heart he was basically in heart failure. His heart was going to die. It was game over for this guy. So it was the choice between game over or go through this procedure, and maybe who knows how long he'll last. The baboons that have received these pig hearts have lasted a maximum of 9 months, but that wasn't because of heart failure. It was actually because of a lung infection, because there has to be the immunosuppressant. And so infections are a bigger problem for animals, people who have these kinds of xenotransplanted organs. So who knows? 9 months? 6 months? 3 years? Nobody has any idea how long this will last. Yeah. This is the first one of its kind. They'd like to do another, and they're going to petition the FDA to do this procedure again. And they are also hoping to get a clinical trial started at some point soon so that they can know whether or not all these genetic modifications are necessary, so that they can know how long somebody might actually have, so that they can get a real idea of how these things work. We'll see in 9 months or whatever.
Between this and 3D printing technology, if we could combine those two things so that nobody has to wait for organs, that would be really nice.
We could 3D print the pig, and then
but that's kind of what I was thinking too, is I know in an ideal world where you can kind of go sci-fi with it and just imagine that 3D printed organs are perfect, that would be the preferred choice. But in the reality of where the science is actually at, the pig heart is the way to go right now.
Yeah, and all of these joking aside, Patrick P. Wonder if he can find truffles now in the chat room. Yeah. All that joking aside, 9 months does not sound like a lot of time to those of us who think time is basically unlimited at the moment.
Yeah.
If when you're up against the clock ticking down the seconds, you're counting the rest of your life in seconds and minutes. 9 months.
Mm-hmm. You can get a lot done. Yeah. It's a gift. It's extra. So good luck to this gentleman. And we wish him well, and we hope he is the first of many, and that many lives can be extended with this technology moving forward. Justin, you got a story?
Oh, gosh, I suppose I do.
Gosh, golly.
Let's see. First human ever. Just got a little bit older. We're talking about Omo 1, whose remains are found in Ethiopia by Leakey in the 60s. He is the oldest known Homo sapien that we have found. That is the atomically modern human. Oldest one we ever found. Until now, we didn't really know how old it was. Scientists have been attempting to accurately date this fossil since they found it. And it was thought to be around 195,000 years old. So they been using chemical fingerprints of volcanic ash layers to try to determine the time it was. Some of that sediment, though, was kind of hard for them to work with initially. Technology being what it was at the time. An international team of scientists led by University of Cambridge has readdressed the age of Omo 1 remains. And now it looks like it is the oldest, again, example, pushing back the age of our species as well. So the new research is saying that this thing's about 230,000, at least 230,000 years old. So it knocked it back 35,000 years at the minimum. So it's definitely older than that. There's nothing new that they found about the bones themselves. It's the volcanic ash the bones were found in, or that it was found below, have been more accurately dated. And that volcanic activity that laid down the ash that's on the layer above Omo 1 are dated to 230,000 years. So
that whole relative dating in the fossil record thing.
Yeah, so the bones are older. We don't have the maximum age on the bones yet, but it's older than that. But that pushes back humans, current modern humans, another 35,000 years.
Which is significant. That's we're talking millions of years, but still 35,000 years is a significant chunk of time. I'm saying that as somebody who has not yet achieved 50. So staring down the barrel of it, but still.
Yeah, and I guess part of the confusion is they had assigned it. It's a very volcanic area. This is Ethiopia. This is Rift Valley. There was a lot of volcanic activity back in the day, and they had assigned that layer of ash to the wrong volcano and wrong volcanic activity. Turns out the chemical fingerprint is a match for a volcano that's more than 400 kilometers away. So it was not the closest one, not the most obvious choice, I suppose.
But when you're talking about ash from a volcano, that gets carried through the air, laid down, so it's not necessarily obvious. That's not going to be as obvious. Yeah.
So that's a major update on human origin history, even older than we thought. And again,
we keep getting older.
Rift Valley has because of the volcanic activity, because of the way that valley is laid out and been moving apart. It's not just that's where human activity was most in the world necessarily at that time, but it is like the ideal place to look for fossils. So it's sort of we keep looking at it like it's the oldest, but it's the oldest we've found because we found it in the easiest place to find such things. So there could be somewhere else hiding even older versions of the current modern human. But again, even then, what is a current modern human with this whole braided stream thing? The debate will continue to, But this is the oldest like us that we have found.
Very cool. I love it. Yeah, I it's going to be the explorers, the adventurers who go into caves and are digging places that we haven't dug before, that are hard to reach, that suddenly are going to make the extraordinary finds, because
As
there
they
are
have
definitely
been,
places.
yeah.
It's places that were habitable and easy to reach, or, you know, nice to live in at one point in time, but maybe not so much right now, right? You have to think about what things were like in certain places at certain times to figure out where the people were. Yeah. That's cool. Very, very cool. Blair, you
Yes.
want to bring us in?
Yes. So I have a very interesting study from Mount Holyoke College in Massachusetts. And this breakthrough study has found that just like humans, female dolphins have a functional clitoris. So this is something apparently that's up for debate through most of the animal kingdom. And part of the reason for that is that the human clitoris wasn't fully described by science until the 1990s. So
I'm just sad I'm sad and disappointed by science and the world right now. Okay.
Yeah, well, we were having a
Continue.
conversation in the pre-show about women representation in scientific studies because of a piece of research that came out this week about from linguists looking at gender representation in language texts. And anyway, point being, females of species are often underrepresented in scientific studies in general, especially in humans, especially in medical research in humans. The women are often left off. And so this isn't that surprising, especially based on, you know, cues in our culture about sex and female pleasure in sex and all these. It's not surprising, right? But so that has spread to our understanding of the animal kingdom. And so now it actually takes a research paper to show that a species has a clitoris. So I just kind of wanted to explain how this happened. So first of all, it makes perfect sense for dolphins to have them because they have sex as part of maintaining social bonds. They've also seen females rubbing each other in that area with their snouts, flippers, flukes. So there is, again, lots of kind of anecdotal reasons for this to make sense, and just kind of understanding social dynamics of dolphins. But they actually had to look at clitorises from deceased dolphins to kind of find the structural proof that there was feeling in these spaces and that it was a functional thing. So they looked at the present shape and configuration of erectile bodies. They looked at nerve fibers, and just like the human clitoris, the dolphin clitoris has a large area of erectile tissue that fills up with blood and has free nerve endings underneath thinner skin than on other parts of their genitalia. So it's there. They can feel. Female pleasure is part of sex in dolphins. And researchers also note that there's been little study of the clitoris and female sexual pleasure in nature as a whole. So what I was talking about before is a good reminder to maybe look at that again. You know, just thinking about hyenas off of the top of my head. The alpha female hyenas will actually have enlarged clitoris that resemble a penis. So they're not that different of structures. So it would be odd for it to not do this. It's whatever. Anyway, point being, they've done some research. Hopefully this will help push more research into the female side of sex in the animal kingdom, and it can kind of bring that into the holistic conversation, so it's not just wholly focused on the other side. But
it seems so how many times have we had conversations about studies that have finally happened looking at animals and the different species in the animal kingdom, and we're like, that seems so obvious.
Yes.
If you talk about dolphins, there's always the conversation about how kind of wild and crazy they are. And they talk about, though, usually the teenage male dolphins being hyper aroused and acting. Girls, women are left out of the conversation across the board. Understanding more about other mammals, about other relatives of ours, other species, and how things work generally is going to help us understand more about ourselves as well. And I mean, understanding more about how the animal kingdom works and why different species work the way they do. Yeah.
I'm also surprised this isn't in the animal corner. Now I'm really intrigued to find out what is going to be in the animal corner.
What is going to be in
the
there?
animal corner bled into the short stories today. There was just too much.
I'm like, how did Dolphin Clitoris not become part of that? And there's all right, no, I can't wait to find out.
Okay. Well, now we're going to move on from the functional clitoris in the dolphin to big giant crater on the moon.
Ooh.
It's a completely logical leap, isn't it? Yes. Well, let's talk about a big old crater on the south pole of the moon that is that's called the Aitken crater. The Aitken crater, we can't see it from where we are, the face of the moon as we see it. If you look down at the the bottom end of the moon, we can see the edges of the crater, but we can't actually see it. The only reason we really know about it is we finally have sent craft up and we've been orbiting and looking at the whole moon. And so we know that this crater is a very large and special feature on the moon. So researchers have been trying to figure out, okay, a big impact like that, what would it have done to the moon? When did it happen? How did it happen? And the fact that it's a giant crater on the moon suggests that it occurred after the molten period of the moon. So there was a period of time where the moon was really, really hot after the big impact that shot the moon out from the Earth and other stuff started glomming in and it turned into the moon. That was its own thing. And there was a magma ocean the moon was just a hot magma body in space. And then it started cooling down, getting a crust. That crust was then bombarded and the Aitken crater came to be. But we also have evidence of volcanic activity, these volcanic mares that are up on the northern part of the moon, almost exactly opposite the Aitken crater. And so there's been this kind of thought of like, are these things related? And so some researchers did simulations. We still have to go look at rocks on the moon, which a Chinese rover might be well placed to be able to do. But we have to They took a look at this simulation and simulated impacts on the moon. They simulated the ways that different sizes and angles of big impacts would have affected the moon. And they realized that there's one particular angle that a giant asteroid would be bombarding at. And through that, it would hit the crust of the moon, break into it, create a crater. It would get blasted to smithereens, but it would go, yes, Blair, into the moon. And the asteroid would just burrow itself right in there. But that would also set off magma movement, material movement through the moon that would lead to the creation of these little volcanoes exactly opposite the impact crater.
That's crazy, and I love it.
So something just punched right into the moon, and the moon went, ouch!
And didn't hit it bad enough to toss it out of orbit.
Exactly. It wasn't such a big impact, a big asteroid or meteor. Yeah, an asteroid.
Do we know? Where maybe the moon used to be somewhere else.
Yeah.
Maybe it had a different orbit.
It was much closer.
Could have been moved.
Or I guess farther. It was like
farther and it got moved in a little bit closer, right? Right. Who knows? I mean, it probably was budged a little bit. Talk about a nudge.
Yeah.
It didn't blow up the moon, but it just added some mass. And so now there might be a big more mass. It just goes to show that the inside of the moon is not just a homogeneous, nice moon inside, that there's a lot of stuff in different places.
So
it's all mixed up and there's been a churn.
And it's lopsided.
And the moon is very likely lopsided. Yes. Yes. But now, thanks to rovers and spacecraft, we have a chance to investigate this further. But like I said, The next step really will be to look at rocks on the northern side of the moon to see what their signatures are like, to see if anything made it all the way through. Because that would be kind of cool.
All
right. That's the moon. So tell me about your little war pony.
I could do that. I was going to stick with the moon theme because I got a moon
you
story
are?
too.
Okay.
Yeah.
Let's go with the moon.
I'm going to stick with the moon, going from moon Churn to moon Chum. And here's my problem with that. A new segment of the show I created just for the following story. But will possibly be using more often in the future as I am getting older and crankier. And I feel like I start more and more sentences with, my problem with that. New research from LSU and the University of Florida suggests that more shark attacks occur on average during fuller moon phases. The results of the study have been published in Correlative Statistical Anomaly Digest, a magazine that allows researchers to publish papers on correlations they have found between no more than two sets of data that have no known causational relationship. Actually, it was published in Frontiers of Marine Science.
All right.
And the authors do point out that they lack a causational link and don't know what the results actually illuminate or if they're even significant, but seem convinced that there is a link there.
Okay.
That said, they do have an abundance of data. Global shark attack record collected over a 55-year period, 1960 to 2015, from the International Shark Attack File, which is housed in the Florida Museum of Natural History at the University of Florida. So shark attacks anywhere, everywhere in the world over a 55-year span, and they found something they think is worth publishing and looking into further. Which is fine. That's what researchers are expected to do. Find a correlation, I guess, of something and look into it further.
I have to say, I just am really enjoying watching Blair's face. For all the podcast listeners right now, you might have heard the sigh that came out of her. But I'm watching her face. And
apparently, Blair has a problem with this too.
Yeah, keep going.
I have so
Justin.
many. I'm formulating a list, but you continue.
Okay. We'll get there. We'll absolutely get there. So the researchers found that more shark attacks than average occur during periods of higher lunar illumination, and fewer attacks than average occur during periods of lower illumination. So over 50, under 50, basically. My problem with that? Using illumination. Most shark attacks happen during daylight. Anywho, so how bright the moon is makes very little difference to a shark underwater in the daytime, which the researchers do clearly state. They say, yes, that doesn't really make sense. Yet then they still suggest that they are in fact seeing shark responses to lunar illumination because they did see a trend. The average attacks in the data do tend to trend toward higher illumination. And this is globally, beaches around the world, consistently, over a long, long time. However, the two highest rates of shark attacks by far happened at the full moon, yes, and at the new moon. That's when there's no illumination whatsoever. Those were the two that had the highest. And the rest of it, it did trend towards, more illumination, more shark attacks. But the two highest were the full moon and the new moon.
Kind of ruins it.
Yeah. It's kind of like when you play Dark Side of the Moon while watching Wizard of Oz. It works.
It works.
Sometimes, sort of. It doesn't most of the time. But even if you're on the right precursor to experiencing that sort of an event, by no means are you supposed to record this and then publish it to anybody of you doing the album with the movie. Okay, so my problem with that, why focus on the sharks at this point? Mm-hmm. Why not the humans? What is it about humans? Right? What is it about humans that sends us running into the ocean at full and new moons? Is missing from the data sets, no matter how many years, no matter how many attacks, this big data record that they got to do something with. The ratio of humans to sharks in the water isn't recorded at all. Yeah, neither
We
are
also know
tides
that full moons make people act wonky.
make
Right,
the people
and so-
act wonky.
And it's interesting. There's also this week there was a meta-analysis published of a bunch of studies, and the researchers suggested that it's not illumination, but it's gravitation. So it's the gravitation that affects the tides, and that gravitational field, even though we can't consciously sense it, impacts the way that animals and plants sleep, move, grow, do things.
Okay.
So you get
Here's my
rid of
problem
the
with that.
illumination. But
Here's my problem with that.
I'm just reporting what I have read.
I know. But here's my problem with that. The new moon and the full moon is the most dramatic tidal changes that take place due to gravitational forces. That's true.
Yes.
But that's not necessarily going to affect a shark either. We're still making that leap of blaming the sharks. That is, however, the 2 times any newbie surfer worth his wax knows it's time to catch the bomb swell.
Right.
It's the full moon and the new moon. That's when you got. So it's not
really great waves.
Not sharks getting aggro. It's more humans in the ocean.
Can I point something else too out? Kiki, put that graph back up, please, if you can,
yes.
if it's not gone.
Nope, I can do that.
So this is what I was thinking and why I was sighing so heavily. Shark attack numbers are not in the hundreds or thousands. So you can say like, oh my God, there was a 20% increase.
Yeah.
The standard deviation on some of these is 1 or 2. So, oh my gosh, there was 1 more shark attack on the new moon. That's tough for me. That's tough to say, okay, this was the highest level at, there were 2 attacks in Australia at the new moon.
Yeah, well, so what this is, though, these aren't, I think what the data plot you've got up there now is deviation from averages, I believe.
Right. And
there's another one that
low.
shows. There's another one that shows more the numbers. This one,
still under 100.
In the beginning, forget the number. The beginning part of this chart is the new moon. The last part of this graph is the full moon, and you can see that's where the peaks are.
Yeah,
and that's also when the most surfers are out. That's also when all of the coastal tidal fisher people, the people who are casting into the waters, that's when they like to go fishing too, because that rapid moving water and that deeper water tends to bring in more fish, which also might be a reason why there might be extra sharks there, but it doesn't matter because even if the number of sharks is always the same, there's definitely more humans in the water, and when there's more humans in the contact with the area where sharks
coming
are
into
causing
contact
confusion.
with sharks, yeah.
There you go. Yeah. So anyway,
yeah.
What's the thing called? Oh yeah. Anyway, that's my problem with that.
Yeah. Very good. Approved.
We approve this message. Awesome. That was a lot of fun. Yeah. So if you don't want to have a higher probability of a shark attack, don't go in the water during the new moon or the full moon.
No, that's the problem. It doesn't matter. No,
I'm kidding.
Just stay out of the water completely. That's the only.
That's
problem.
also the wrong
that
thing to
either.
say.
But there's also like, this is scientific methody wise. There is like, okay, you have to start with some kind of knowledge, and there is some sort of knowledge about fish feeding or mating behaviors under moonlight, or there is some stuff for them to grab onto. But part of the problem is when you get to asking the question and you've already got your data set and you basically have to ask a question off the data that you already have available, you kind of limit actually having a very informed question that you can ask, because you're not doing the experiment. It's like the experiment's already been done. What can we make out of it?
Right.
Here's the data. What can we infer from this? And that's a tougher one.
It's post hoc.
Scientific method.
Yeah. Exactly. I mean, this is one of the big arguments about genome-wide association studies and about like using machine learning to pull results out of big data sets. It's okay. The relationships are there already. How do you know you're getting the right one?
And literally, if you just took the phases of the moon and any other data report, car accidents, children being born, crimes being committed, you will find that one phase of the moon or another has an average different than the others because that's how numbers work.
Yeah.
Yep.
That's just how numbers work. Not everything happens evenly all the time. It just doesn't. So if you took 55 years of new moons and full moons and everything else, you're going to see a statistical average difference in those phases that has nothing to do with the moon.
I like it. Quick story before we move into our COVID update. The zoo. Blair, this one is for you. If you step into the zoo and you take a big inhale through the nose, smell, how many animals do you think you could identify just from
one? There's usually one that hits you right across the face when you walk in.
Well, researchers publishing in Current Biology this week have published their work. 2 different groups, and it's really great because both these groups were working on air DNA, the use of identifying DNA in the air to identify species of animals separately. But then they heard about each other's work, and so then they decided to publish their work at the same time in the same journal. So it's like more replications. More better science. And so this is when science really starts to work. They put their setups, their DNA sniffers, in zoos because they figured, logically, if you went to a farm, you could sniff cow DNA, but how do you know if that cow DNA is like from that farm or from some farm somewhere nearby that just got blown there on the wind? If you're sniffing tiger DNA, you're probably only getting it from the zoo. So
hopefully,
to limit their area and the identification, the zoos gave them this awesome environment from which to sample DNA. And they were able to identify the animals in the zoo. So they basically used like a computer cooling fan with an air filter attached to it and then let it run. And that air filter then basically collected all the DNA, and then they were able to wash it and do all the DNA sampling from there. And the different studies, they were able to identify over 25 species of mammals and birds, DNA belonging to an Eurasian hedgehog, which is endangered in the UK. This was a study at Queen Mary University of London. A team in Copenhagen detected 49 non-human vertebrate species, mammal, bird, reptile, amphibian, and fish. These were animals like the okapi, armadillo, even the guppy in a pond in the tropical house.
Yeah. That's awesome.
That's so great. And they got squirrels and rats and house mice and other fish species that were used as animal feed in the air also. And they they really double checked and overchecked to make sure contamination wasn't an issue. But they were pretty confident that they were sampling species who live at the zoos through the air. And so why is this a cool idea? Well, you don't always have zoos. You don't always know what animals are in different places. So for cave ecosystems or jungles, or think about just about anywhere, even in urban environments, you could sniff the DNA in the air and potentially identify animals and maybe find animals that we've thought to be extinct.
Mm-hmm.
Who knows?
The first thing I thought of is like, this looks like thylacine DNA.
Yeah.
Oh, I was thinking, I immediately went to the Borneo Kalimantan monkey cat.
Yes.
An elusive creature that's been evading jungle cameras for decades now.
We will get the monkey cat. I love it. Yeah. So not just a DNA from the cells, right? Things that you leave in the dust and the dirt or in the water, but the air, which is I think, pretty amazing. I think it's just pretty awesome. All right. This is This Week in Science. Thank you for listening to the show. We appreciate you being here with us. If you are enjoying it, please share it with a friend today. All right, we're going to come on back right now, and we will be giving our COVID update. So, Blair, you have a fun story.
Yes, it's a fun one. It's also extremely brief and simple. It's just that in previous studies, researchers have shown that T cells induced by other coronaviruses can recognize SARS-CoV-2, COVID-19. And this new study examines for the first time how the presence of those same T cells at the time of SARS-CoV-2 exposure influences whether you become infected. So that would include T cells that you have due to infection from the common cold. So that is a coronavirus, right? So this provides a blueprint for a second-generation universal vaccine that could prevent infection from current and future SARS-CoV-2 variants, including Omicron, because these T cells protect from COVID a little bit differently. Instead of attaching, going to the spike protein, they actually target internal proteins within the SARS-CoV-2 virus. So they're pretty much good against all the current and hopefully future variants. So this is a small sample study, 52 people. They were all white and European. So again, very
Limited.
Preliminary.
Yeah.
Lots of variables that need to be tested here. But it would appear that there is something to be learned here. This does not mean you are safe if you got the common cold from COVID. What this means is the methodology that these T cells use to try to protect you from other coronaviruses could be used to make other vaccines, which would be great.
Yeah, hopefully it doesn't mean that the cold protects you, because you know what that would mean then? It would mean that all the people who never get colds because they wash their hands all the time and are very sanitary and avoid sick people and are really good at it are the people who are dying. And what we're going to be left with is a population of non-hand washers who always catch colds.
Hey, listen, I wash my hands very diligently and I get sick multiple times a year, every year. Maybe that's why I haven't caught COVID yet.
It's possible. I mean, there's also it is winter, and if you are out and you've gotten colds because you are not a hand washer, you know, your body's immune system probably is elevating a temperature, getting some other immune cells activated. You have an inflammatory response that possibly could help to protect you as well. I mean, I'm just looking on the bright side here. I don't know.
Rick Loveman, yeah, you don't want a cure for the common cold. No, if we cured the common cold, 20 years ago, we'd all be in big trouble.
Oh, boy.
That's awesome. Justin, what was your story?
I was just good news.
Woohoo!
COVID edition.
Awesome. I'm ready.
Oregon State University researchers identified a potential COVID blocker in hemp because, of course, they did.
Bump. Thank you, Oregon.
As much as it sounds like a making-up story, actually, this looks pretty promising. Findings of the study, led by Richard Van Bremen, a researcher with Oregon State's Global Hemp Innovation Center, were published in Journal of Natural Products. Okay, so far, maybe. But they looked at hemp. This is cannabis sativa. And they found a pair of cannabinoid acids within the hemp plant that bind to SARS-CoV-2 spike protein. So it actually attaches itself to the spike protein of the virus. So that blocks a critical step in the process the virus uses to infect people. So the compounds are cannabigurolic acid, CBGA, and cannabidolic acid, CBDA.
Yeah, cannabidurolic, cannabidiolic. Yeah.
The spike protein is the same one that is used as the drug target, same drug targets used in the vaccines and in the antibody therapy. And as the report points out, a drug target is any molecule that is critical to the process of the disease progressing.
Yeah.
Disrupt that one molecule if it's critical, and the disease cannot progress. In this case, these CBD precursors is basically what these are. Do that job. It's so funny though. It's like multiple times in this story, Bremen is going on saying, these are not the controlled substances like THC, the psychoactive ingredient in marijuana. The thing researchers are constantly having to explain whenever they're talking about using hemp for anything. And then later on, they explained that it's not CBD itself, but these are CBD molecular precursors, chemical precursors. So yes, you're safe. You won't feel mildly relaxed. You can go on being stressed out all day.
Yeah.
So the interesting aspect of this. So right, this is just looking at molecules. These molecules that happen to be coming from hemp interact with the spike protein. Awesome, but we don't know whether this is going to work in a human person. I mean, if you enjoy your CBD gummies and you enjoy all those things, then fine, that's great. But don't think that means you are protected against SARS-CoV-2. This is a study that has only shown affinity for the spike protein by these molecules. It is not showing that it's going to keep you safe. This isn't a treatment. This is not this is the earliest stages of research. So we need to say that because people are already just like, woo! Yeah. We can't let this go down the bleach or the other roads.
And this is one that they have tested that does work on the spike protein. They were looking at another compound in licorice that they haven't tested, but they think it will have similar properties. But I think one of the things that I found very interesting about this, these are precursors that are in some abundance in hemp. We did a story a couple years, maybe 2 years ago, about a researcher that was able to produce CBD via I believe he used yeast, in a bioreactor. And one of the things that was interesting about that story then is because they're talking about, this is part of research, they're trying to use CBD to do maybe as a concussion protocol because it has some neuroregenerative properties. Something about CBD and the cannabinoids, they interact very well with the human system. Whether humans have been interacting with hemp for a long time or hemp just happens to be a plant that interacts with human physiology very well, I don't know. But one of the things that was interesting in that is the many, many hundreds of other CBD-like compounds, precursors and the rest, that are produced in much smaller quantities within hemp that don't have a large enough effect or placement that can actually be expressed in that biotechy way and then researched. Here's a pretty good example of now going back a step from CBD that we've already kind of know has some uses, a precursor that could be utilized as medicine as well. This is a plant that is as much as it's talked about sometimes for the right reasons, sometimes feared for the wrong reasons, and the right reason, all that. There's a lot of research that needs to be done still onto this plant and the compound.
Yeah, a bunch of research. I mean, it looks interesting. It's promising, possibly, but this is not in humans. This was just in cells. More research needs to be done before people really dive into it as prevention and treatment for SARS-CoV-2. So I just want to say that over and over and over again because it's great cell study. I mean, it's like when we talk about all sorts of other stuff and then we're like, oh, in mice.
Maybe it'll be ready for the next coronavirus.
Yeah, maybe it'll be ready for the next one. Who knows? I mean, this is like licorice, the CBD variants. Like, let's see. Yeah, more research. Let's put more money to it. I know that with the Farm Bill, hemp has a lot more research going on right now. So that in itself is cool. Oh my goodness! This is This Week in Science. Thank you once again for joining us for this episode of our weekly science news talk show. If you would like to help support This Week in Science, you can head over to TWIS.org, click on our Patreon link, and choose your level of support. We have many levels to fit different budgets, $10 and more a month, and you will be thanked by name at the end of the show. I can't wait to read your name. I love doing that, and I love having that list of names and getting to share it with everybody. So I do hope that you will become a part of keeping this show going. We thank you for your support. We really can't do it without you. And now it is time to come back into the more animalistic part of the show we know and love. Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
I have a story about a dog. This is actually
dog?
Yes,
Named
the dog.
Sadie?
No, this is a dog
okay.
named Kun Kun.
Okay.
This dog is owned by Laura V. Kuaya, who's the first author on this new study I'm reporting on today. She moved from Mexico to Hungary to join the Neuroethology of Communication Lab at the Department of Ethology at TWIS Lorand University. And so when she did that, she wondered whether Kun Kun noticed that people in Budapest spoke a different language than the people in Mexico. We know from previous research that people, and this includes pre-verbal human infants, notice the difference between languages fundamentally in their brain. But we don't know if dogs know the difference between languages just by the fact that they sound different. So she designed a brain imaging study to see what was going on in Kun Kun's brain. Kun Kun and 17 other dogs were trained to lay motionless in a brain scanner while wearing headphones.
Oh, what good doggies. Oh, what a very good boy.
They played speech excerpts of The Little Prince in Spanish and then in Hungarian. All dogs heard only one of the two languages from their owners previously, so one was familiar and one was not. They also played scrambled versions of those excerpts, which I'm sure sounded nightmarish, which sound completely unnatural and nonsensical, to test whether they could detect a difference between speech and what they would consider non-speech. What they found was that there were distinct activity patterns in dogs' primary auditory cortex if they were listening to speech versus non-speech. They could tell right away if it was real language, if it was muddled up sounds. This distinction was independent from whether that stimuli originated from the familiar or the unfamiliar language. So when they paired it with the one they were used to or not, they could still tell the difference between speech and non-speech, even if they didn't speak Hungarian. So that's step one. But they could also tell the difference between Spanish and Hungarian. They found language-specific activity patterns in their brains in another region, which is the secondary auditory cortex. But the older the dog was, the better they were at distinguishing between familiar and unfamiliar language based on their brain activity. So this suggests that during the time that dogs live with us, they pick up more and more on auditory regularities of the language that they hear every day. And so they are able to distinguish, okay, that's definitely speech, but it's totally unfamiliar and different. And they're able to distinguish that, I don't know what's happening. It's not language, though. So I'm
not surprised that dogs can figure out the difference between speech and non-speech. I mean, we've talked so many times about how attuned dogs are to human communication, to human wants, needs, the ability to stay with their human as a member of the pack, to take instruction, that there's definitely the learning of certain words and certain signals.
So there's a couple things about this study I want to point out. So One of them is the potential implications, but then I kind of want to poke a hole in the study just for fun, because that's what we do here, right?
Yes.
But so the implications here are a non-human can tell the difference between languages. Can we do that the opposite way? Not without computer modeling. When we found out that Hyraxes have different, what did they have? They had different dialects depending on where they lived. We couldn't tell that without using a computer. We couldn't tell that just by our naked ear, that the Hyraxes had a different accent. I lost language here while we're talking about language. The language center of my brain just shut down completely.
Language goes.
They have a different accent or a different way of talking. So this is something that we can't do. I can say with quite a bit of confidence. We know chimps, for example, have different quote unquote languages depending on where they grew up, and we can't tell that without computer modeling. But to
be fair. There's not a lot of humans that were raised by these chimps.
Right. Sure.
Who've been hearing them every day. Who then might,
So this is the second piece of that. Is this dog specific? Is it domesticated animal specific? Is it across the animal kingdom? Can they tell different human languages? So there's a lot of interesting questions. Now here is the hole I want to poke though in this study. All of the pictures I saw of this study had some sort of border collie type dog. Now, these dogs are not coincidentally very smart and trainable, and therefore they could be trained to wear a set of headphones and sit in an MRI machine. My dog could not do that. But they're also famous for knowing 200 words, border collies, some of them.
They're like the dog version of Alex the parrot.
Right. You can say, go get Einstein, and they'll come back with their Einstein doll. So I think this could be very specific to the subset of dogs that were in this study, because they are already extremely smart, extremely trainable, extremely focused on humans for those reasons. And on top of that, can learn the most human words out of pretty much all of the breeds. So there is a weird accidental Venn diagram of like dogs that you could teach to sit in an MRI machine and dogs that can understand human language well, right? So if there was a way we could parse that out, I feel like this would be more accurate.
Just looking at the study very quickly, their first table in the study names the breeds of the dogs. And from the 19 animals, we have Labradoodle, a mix, Golden Retriever, Border Collie, Cocker Spaniel, Australian Shepherd, and those are the breeds of dogs, which are all fairly trainable and intelligent,
smart and human focused too. Yeah, very human focused. So I think this is very cool, very interesting, awesome to hear. Certainly something that we can't do in reverse.
But
we can't
it could
tell
be
between barking in Spain versus barking
but it could be biased
South Africa
because of the types of dogs we're looking at.
No, absolutely. Yeah.
Interesting. So now we just have to get the dogs that don't care about people to see if we can train them.
Or they could just care,
dogs.
But like they could care, but also just not really be that interested in doing an experiment.
Right.
If you could find a way to fit a hat on Sadie that she couldn't shake off, and that would give you brain readings, then I would believe it.
Oh my gosh, I'm imagining a helmet on Sadie, and she'll look like Vince Clotho from Ghostbusters. Never mind.
Anyway, speaking of helmets, let's talk about hats. Or perhaps pants or sleeping bags. I don't know. So this is a story about orb weavers from Australia. This is the leaf curling spider. It is Phononatha griffii, and they are a type of orb weaver that is known for curling leaves into their webs. They use that leaf, they shimmy into it with their abdomen. Oh, there we go. So they shimmy into it, and they still have their legs sticking out, touching their webs, so they can tell what's going on around them. They can see, they can sense. But that leaf serves a really important purpose. It actually protects them from predators like birds. They are hidden in it. And it also shields them from parasitic wasps, which are famous for laying eggs on the bodies of other insects and spiders, which kill their hosts. So they're protecting themselves. They actually use spider silk to lift a leaf up from the ground into their web. They put it in the middle of their orb web, and then they curl it up and secure it with silk. They roll it up like a sleeping bag, and then they shimmy on in. And so this is a really interesting study because they only leave that little leaf to eat, really. But what I found even more interesting is that they will actually shack up with their partner. So a lot of spiders we've talked about on the show, the males show up to copulate with the female, and that's it. They're lucky to get away with their lives, often not with their genitals, but their lives for the time being. Sometimes they're eaten. It's a whole thing. But in this case, a lot of the time, males and females will form pairs and share their little leaf sleeping bag. The males will move in with the females when she's still young. That's how they do it without getting eaten. And then once she's mature, they'll mate with her. But they still do sometimes cannibalize the males. Of course, it wouldn't be a spider without that. That occurs independently of whether the female has been deprived of food or not. So it's just kind of if she feels like it, if he's bugging her. But after mating, the female actually makes another curled leaf retreat in vegetation away from the web. And this is her nursery little leaf. So that's where she lays her eggs.
Talk about nesting. I love it.
Yeah. So they're nesting. They're adapting their environment around them to respond to threats. This is, in my opinion, 100% some real tool use being used by these spiders in their webs to augment their webs with something more than the webbing that they create. And so I love this story. I think it's so fun that the orb weavers are like, I'm kind of exposed out here. Oh, there's a leaf down there. What if I just curl it up around myself? I'll be out there. I can still tell when stuff's going on. I'm not going to block my face, but I'll be protected. It's a nice coat or a hat. Or what do you think? If it's on their abdomen, what do you think it is? Is it pants?
It's a skirt.
I think I would go further. I would call it shelter. I would call it shelter, and I would say that might mean that orb weavers invented shelter, depending on how long they've been doing this, before humans.
Yeah,
we go run into a cave, but building your own shelter.
Yeah, they're the original campers.
Yeah,
pitch their little tent and.
But you described it as a lot of things. It sounds like it's partly armor from getting eggs laid in you while you sleep or something horrible happening to you.
Yeah. The other interesting thing is that when they're younger, so orb weavers only live a year, by the way. Oh. But when they're younger, if they're not strong and big enough to pick up a big dried leaf, they'll use a fresh, smaller leaf, or sometimes they'll use snail shells or a piece of paper.
Oh.
So if you see a piece of what you assume is trash in a web, might actually be a little spider living in there.
So
I'd love to know if it's more species.
Adaptive ability too.
Mm-hmm.
Yeah, it's interesting. I mean, using. I mean, this is tool use, right? If it's used for a purpose, is it instinctual? Is it, when does it happen? I mean, you've talked before about the complexity, the chaotic nature of spider mating, of the males spiraling into the females, and how order comes from that. Is this one of those things, or is it? Is this spider planning? Is this something that worked a lot at one point and then it's just ingrained now because it just worked really well? Do other spider species do this beside these Australian spiders?
And you bring up a good point there too about whether or not this is instinct. So I don't know that I don't know that spiders go to a spider engineering university to learn how to make their webs, but they all do it.
Yeah.
So there's something more than just instinct to have web instinct to have shelter or to wrap things. There is genetic memory somehow being passed down on how to do things.
There is, but there's also adaptive learning throughout their life. So the kind of orb weavers that we have here in my area, some of those are the ones that take down their web every night and put it back up in the morning, so it's so clean that bugs can't see it and they walk right into it. They also do it usually at face level, and it's so clean that you can't see it, which is the worst.
The bane of Blair's existence.
One day we'll catch one of those humans, and when we do, we will eat forever.
Oh man, I mean, orb weavers they're not even venomous to humans. But anyway, the crazy thing about that is if you knock one down, if it's like, get out, this is right in my face, and you get a stick and you like nicely move the spider, very important piece, and then you knock down their web, they won't build it there again. They will build it somewhere else. And in this case, they change what types of material they use to make their leaf depending on what they are capable of lifting and what is available. So there's adaptation happening here within their lifespan where they know they're trying to get themselves, it's pretty clear they're getting to a goal. I don't know if it's instincts or something else that's telling them they need to get to that goal, but the fact that they can adapt their procedures to get to where they're going, to get themselves their little shelter, that means there's something more than just, I put leaf in web. There's something more going on.
Yeah, and it's their little bit of shelter, but at the same time, maybe that little bit of leaf or whatever could also be an attractant to an insect or something. I mean, I don't know. Maybe it also helps them eat. They're hiding in there. The insect is like, ooh, what's that? And then they're like, ha, I got you.
Yeah.
I got you.
Yeah.
Oh, spiders. The more we learn about the spiders, the more I love them, the more I appreciate all the spiders of the world. I still don't know if I will go into the basement of my childhood home where all the black widow spiders live under the stairs, but I still appreciate them a lot. All right, Justin, it's your turn. What do you have for us now? War ponies?
Let's do it. So medieval times evoke a number of colorful images and imaginings. Mostly of filthy peasants working fields and a general lack of hygiene and scientific understanding, but also of the brave knight, resplendent in his shiny armor, sitting high upon his trusty steed. The mighty warhorse, too resplendent in armor perhaps, a beast of good breeding and powerful athleticism, the very essence and envy of what all horses aspire to be. And while huge sums were spent on breeding and training and keeping horses for combat, as much for actual warfare as for local symbols of power and authority, turns out medieval war horses, as you were leading into there, were no more than pony-sized. New study is showing horses during the medieval period just weren't big. We're not that big yet, at least. Team of archaeologists and historians have found that they were not even bred for size. That wasn't even the thing that they were shooting for. Back then, they were just the success for inbreeding was for things like temperament. They wanted long distance
fierceness,
rating. They'd be in tournaments, but they had to they just wanted horses that would listen. That was like apparently the high bar. And also maybe the knights weren't as tall.
I was gonna say, I wonder if people were not as heavy. Because tall, but also the thing I'd be really thinking about would be how heavy. Because especially people are wearing armor as well.
Yeah. So then picture a very robust pony, right? Like this is like the knight is riding into battle with just inches off the ground, probably lifting them up so as not to drag in the dirt. This just totally changes the picture of how they have to be portrayed from now on in every movie. You can have no, and even the tallest, the biggest horse they ever found, and this is from an era of 300. The biggest horse they found, it would be considered a small riding horse today, and that's the biggest one ever found in the records from then. But yeah, they were focused on the temperament, just getting horses to pay attention. That's all they really wanted. Knights on ponies. Keep that image. Make sure that's if you see any of those historical documentary reenactment things, make sure they're not using big horses.
So nobody had looked at the bones before to really check and see how big these war horses were? Because we've got stories of we look at the horses now and say, these are the breeds that were the war horses of the past. And
well, no, what they do is they put the biggest horse out there. That's the war horse. It's the big, powerful, strong horse with a tall knight on it and everything like that.
Yeah.
But, when they specifically went back and looked for horses that were being bred anywhere, really, but specifically the ones that were being part of breeding programs that would have been used for combat and trained for combat. Yeah, not that big. Not that big. Very small horses, in fact. Yeah, ponies.
Well, they were small, small in stature, but maybe stocky, so that they could have the weight of the armor, like Blair
maybe
had
not.
said. So maybe little
they
stocky
just panted
ponies.
Heavily.
Panting, stocky ponies.
So the image that we have of the warhorse did eventually come about in the late 1600s, at a time when getting into the time where they started to become more ornamental. Where there was a period of time there where they may have actually, really been used as war horses for like a little while. But by the time you got the big horses, it was over with. The knights riding into battle on horseback, that wasn't a thing.
Nobody was doing that anymore. Awesome.
But yeah, serious question about it, and you're right. Yeah, average human height was also smaller back then. Those large horses, if they had them, if it hadn't, it's just been too hard to get on.
I would think in battle, it would be a strategic advantage to be higher up.
Yeah, but also
than infantry that's on foot.
And other than that, it was all infantry, right? So that was the other thing, is just having a horse and being able to go places quickly, or send your probably used more internally than they were for an external war, in terms of those filthy peasants working your farm day and night. It's not because they necessarily wanted to be there; they didn't want to get run through with a lance if they didn't go to work that day. So there was a lot of other dynamics. But yeah, that was a huge. So that's going on over there in the UK. Now, around the same time period, 500 to 1100, the highlands of Peru were home to the Wari people. The Wari culture is spread over vast distances, the rugged terrain of the Andes Mountains, and long, huge territory the Wari controlled. This is before the Incas even existed. New finds from a small site in Peru suggest these Wari culture may have forged their political alliances and authority by serving drug-laced beer to the local elites in other villages.
That's how you make friends, huh?
Yeah.
Hey, I'm new to the neighborhood. Want to come over, have some drinks, some dinner.
Disclaimer: do not do that at home.
No. Take back that disclaimer. If you are a government, and instead of sending the military to go fight another country, maybe send them your best drugs. Maybe hey,
as long as they know they're drinking
party
drugs
instead. Yeah. No, they weren't so a great question, because it's drug-laced beer, right?
Yeah.
So that is an important point, because the drug is a jungle drug that didn't grow right. One of the places was 800 kilometers from their capital. They found this outpost. That's where some of this research was done. The drug that they're bringing with them is something that's found deep in the jungles and places far, far away from where this little outpost is. It's something that you can pulverize into a powder. It gives a very strong hallucinogenic out-of-body type experience. It's a very intense drug. Turns out, when you mix this drug with beer, which the locals were making, it has a much milder calming sort of effect where it's more like a party thing. Everybody feels good and wants to socialize and talk and feels nice. So basically what they found was that the outpost, only inside the Wari's little small outpost, which could have had no more than 100 people in this larger region, was all of the crushed seeds from this hallucinogenic drug. Outside of that was where the local villagers did their brewing, and they would find the combinations of this drug and the alcohol. So they would have people come over to specifically do the drug with the alcohol with them at these parties. Now, fantastic. I love that story. Part of what I really love about this story, this is like, I think the first time going back into the record, I've heard about drugs being used for any form of recreation. They always are going like, oh,
religious ceremony.
Ancients, it had to be a religious ritual ceremony because all those people did was be in awe of deities, and then, whatever else they did all day, who cares? They just did ritual worship stuff. No, they partied with the foreigners, and they established trade routes based on that and goodwill and everything else. And it's interesting too, because that village, 800 kilometers away, took on all of the cultural adornments of the Wari people, the pottery, the housing styles, all of that. But there was no evidence of enslavement or warfare or bloodshed around it, just the arrival of the cocktails. That's all it took.
We don't need to fight. We don't need to fight. I've got a better idea.
Maybe this is why Americans cling to the idea of quote unquote the first Thanksgiving so hard, because if that's actually how it had gone, things might have gone much better.
A bit better.
Just would have shared some nice food and drink with your new neighbors. But no.
Yeah, would have been nice.
Food and drugged drink.
Yeah.
Okay. little hallucination with your new friends.
Yeah.
Oh, don't mind. As we dig back in the archaeological record, I'm sure we'll find more evidence of non-ceremonial uses. There's going to be a lot more going on.
And there actually is within the Wari people. They have a lot of artwork on pottery and other things showing the seed pods from this Vilca plant, as well as sort of festivities around it. So they definitely had a party culture and ran an empire of partying like that. Why? When's the last time you've seen this? When's the last time this is?
I don't know. College, maybe.
Yeah, I was going to say Greek row.
We're almost to the end of the show here. I have a couple more stories. And I'm so excited about these stories. Let's talk about fingerprints. I want you to look at your fingers and look at your fingerprints, and you've probably got some little ridges if you haven't burned all your fingerprints off accidentally doing, I don't know, whatever. But
how about Martin scars?
Right. Martin scars, whatever. You may have lost some fingerprints depending upon your age, but each and every one of us has unique fingerprints. It's why you can get fingerprinted and put your fingerprints on record so people can identify you. It's this very special, unique marker.
Ish.
And what?
Unique-ish. I mean, there's like they can be pretty similar, actually.
They can be very similar. But
they're not as unique as people think.
They are very unique. With other, they can be used to identify people. So we've got the radial loops, we've got arches, there are whorls, and these different patterns. People have wondered where they come from.
Yeah.
For a very long time. Yeah, how do they come about? They come about during embryonic development. They start to form about the 3rd month in utero, and they develop and change as the baby is growing. And so they are definitely genetically linked, but which genes control them? Well, some researchers in China decided to take a look at it, and as they dug in to identify the genes that were responsible for fingerprints, they found that they are also, there were a number of genes. One in particular, these is related to limb development. So as the limbs grow, the fingerprints get their shapes. And those loops and arches and whorls, the researchers think might be from the impact of how the growth takes place. And so maybe as the bones grow and push outward, that pressure from the growth of the bones influences the skin and how these ridges and whorls and loops form.
So I never realized, I'm looking at my own fingers right now. I never realized, is that normal that you have kind of, you showed us these 6 different kinds of types of fingerprints you could have, that you have the same across all your fingers usually?
You don't necessarily have the same across all your fingers. No. They can be different. It depends. But they have found very specific patterns, which is kind of cool. So they have discovered that there are particular things. People with whorls in their fingerprints on both of the little fingers tend to have longer little fingers than people who do not. And this correlation is also linked to the genes involved with limb development. Yeah, so there are very specific patterns that tend to pop up more or less frequently, depending. And they think that it may be force related. Like I mentioned, that as the growth takes place, everything stretches and elongates, and there's pressure that's involved from that growth, and that might change what the shapes of the fingerprints look like. It might actually influence them. And so that might be why it happens. But there are also congenital disorders that are linked to what they call dermatoglyphic patterns. This is the new word of the day, dermatoglyphic. Fingerprints, or the lines in your palms. Those are dermatoglyphs. So children with Down syndrome are likely to have a single crease running across the palm of their hands. But this could also be affected by developmental genes. And so there's a theoretical basis for this kind of pleiotropy, the researchers say. Pleiotropy being where you have multiple traits affected by similar genes, by the same genes.
You know what I've noticed is that the older that I've gotten, my fingerprints have vanished. Kind of right along with my eyesight.
Well, that's what I was going to say. I think I need to go get my glasses so that I can actually see my fingerprints now.
Because I used to have them, and I'm wearing glasses, but I don't know where they went, because they're just gone. I used to be able to look down and see them. They're not there anymore. I mean, without the glasses, I barely have hands.
Well, maybe you don't get to have fingerprints anymore because we're in the shrinking stage. I don't know. No, not yet.
Oh my gosh.
Not yet.
That's fascinating. I want to learn more about fingerprints.
Fingerprints are fascinating.
I've never really looked at mine, and now I'm very
fingerprint focused.
It's really interesting.
Should I have so many creases in my fingerprints?
I have a lot of creases.
Okay, I've got a ton now. Oh my goodness.
I love it.
I can't see.
I hope everybody is, except if you're driving, staring at your fingers.
Wait until a red light, then you can.
Please keep your eyes on the road. Last story for the night. There is some new research into CAR T therapies. So CAR T therapy is therapy where normally T cells, which are immune cells, as we know, get removed from the body and engineered to express a receptor, the CAR receptor. Now, the CAR receptor then identifies or latches onto markers on the outside of cancer cells. And so then CAR T cells, the T cells, because they're attached to that receptor that then is attached to a cancer cell, can destroy the cancer cells. And so this is how the CAR T therapy, through this genetic engineering, enables the treatment of some cancers.
Mm-hmm.
This last week in Science, researchers have published their work in which they no longer have to take cells out of the body to engineer them to express the CAR protein. They, in their work in mice, this is once again in mice, and so in working in mice, they, from the University of Pennsylvania, they were able to get these CAR cells produced in the body using mRNA, like the Moderna and Pfizer BioNTech vaccines. They injected mRNA to get the T cells in the mice to produce the chimeric antigen receptor, the CAR receptor.
Wow.
And this CAR receptor, what they had it do in this particular study was target the it's a fibroblast activating protein. And the fibroblast activating protein, what they did is this in scar tissue formation, fibroblasts, this activating protein is stimulated. This is a problem in cardiac disease. If you have high blood pressure and problems with the heart, which they made a model of this in the mice. So they gave mice heart problems. The mice started to produce these fibrotic tissue in the heart, scar tissue with this fibroblast activating protein. They did the mRNA treatment. The CAR T cells then latched on to the scar tissue, reduced the scar tissue in the hearts to the point that they were just about like normal hearts. Now, the cool thing about this, if you do the take the immune cells out of the body and then put them back into the body procedure, you end up with these T cells, these CAR T cells running around the body, and they're there all the time. And this can actually be a problem. And researchers would like it to be more transient. And so with the mRNA, it produces a transient response. And they showed that these CAR T cells, what they're calling FAP CAR positive T cells in mice, they were pretty much gone about a week after injection. They didn't see any more expression. Now, there are still a bunch of questions as to how this could work in people, but it could be used not only for this fibroblast activating protein, it can be used to attack cancers, it can be used to attack all sorts of things and to develop just about any novel therapy that you could probably think of. So, Justin, Justin, you've been talking about this CAR T stuff for a really long time, and I just think like this is the next step that is going to take it to the next level. And we're starting to look at it as like, this is going to make a real difference in people's lives combined with mRNA.
I mean, it's already been shown to be able to knock out leukemia in a patient within 48 hours. And persist in that case in a way that prevents them from getting it again. One of the things is that there was a percentage of people who would just die from it, from undergoing that procedure. The other part of it was that it cost $2 million for one person to undergo that treatment. And that had to be, that was taking cells out, re-engineering them, like with a shelled out HIV, I think it was, and then reintroducing.
Right.
So
and so I was going to say it's limited in location because not every hospital or treatment center is going to have all the equipment that would be necessary for the procedure. And so it's almost like, the mRNA vaccine. There's your vaccine. You'd maybe come in and have an injection.
And you're coming into it's not even this facility that's even, you've got a facility, that one facility that can do these treatments, can now package it,
anywhere.
Send it anywhere. Oh, this is huge. Very good.
And if you can think about anything, that you like any aberrant, what the researchers say, one of the researchers says it's groundbreaking because it's a whole new way of thinking about a therapeutic application, redirecting the T cell to control other aberrant cells.
Mhm.
So we think of, cancer cells as being aberrant, but like what other cells, what other things are potentially going wrong that in the body at different points in time that could be
and that's
old.
And that's also the thing about the leukemia was that leukemia is white blood cells that have become cancerous. There's slightly different. So you've trained white blood cells, T cells, to attack white blood cells that are have infected or built wrong or however you want to put it, that are cancerous. In that they're being produced and that they're non-effective. So it was able to identify, so it can be very strategically targeted. Fantastic to find out they've got a new, better, cheaper, faster method available in mice.
In mice. Yes, in mice. And then that'll be the next step. Can we use this in people and will it help? So it's very exciting. I think we will hear a lot more about this probably in the not too far off future. But the future is coming. It's here. The future that you didn't want to wait for. The end of the show.
Oh.
And we made it to the end of the show.
We did
I
it.
think we might have.
We did it. We made it to the end of the show. Happy anniversary, Blair.
Oh, right. I forgot about that. Thanks. Well, I'm glad I brought all the animal stories today then. That's
you brought the spiders, you brought sex, you brought it all.
Dogs and
dogs.
MRI machines, always good.
Dogs. Yes. Oh. It's very, very exciting to have you be part of the show and have you being part of the show for such a long time now. And the show would not be this great without you.
Well, thanks. I wouldn't be the same without this show, so I appreciate that as well.
Yeah, we appreciate you, Blair, so much. So everyone, if you can take some time to wish Blair happy anniversary. It is her 10th anniversary here on
10
TWIS.
years.
She's been with us.
Oh my goodness.
10 years talking about animals, invertebrate sex, all the fun stuff.
Wow.
Spiders, poop.
Where do we get you out of high school? 10 years.
Yeah, I wish.
Almost.
Yeah, it probably wasn't that far away, huh?
That close. Everyone, thank you for joining us for this episode. We hope that you've been around for a long time enjoying Blair's additions to the show. We hope that you will be around for many years to come to continue enjoying Blair's additions to the show. And we're very glad just to have you here with us tonight. So thank you all for another episode, and thank you. There's time for some shout outs to people. Thank you, Fada. Thank you for your help in doing show notes and what else? Oh, social media. That's the big one. That is the big one. Thank you so much for that. And Gord and Arnlore, thank you for your work keeping things decent in the chat rooms. Identity 4, thank you for recording the show. And Rachel, thank you so much for your editing and assistance. I do appreciate it. And to all of you patrons, thank you for your support on Patreon. Oh, I got thanks for you. Thank you to Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vigard Chefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Don Mundus, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles Jack, Brian Carrington, Matt Bass, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Romney Day, Flying Out, Christopher Dreyer, Artyom, Greg Brid, Greg Briggs, John Atwood, Hey Arizona, support Aaron Lieberman for governor. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Old, Dave Neighbor, Eric Knapp, EO, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, head over to twist.org and click on the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and our Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe while you work in your garden outside, trim some leaves, pull some weeds. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
For more information on anything you've heard here today, notes to the stories and links to them will be available on our website, www.twist.org, where you can also sign up for a newsletter.
You can also still buy calendars there if you don't have a calendar, right?
Oh, I know.
Or are we all out? No,
we're not all out yet. Very
We're
close.
not all out yet. So you can definitely buy a calendar. It's still
Yep.
January. There's still time. You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be spam filtered into an audio recording that is piped into headphones on the head of a dog, and they'll be able to tell whether it's in English or another language, but they won't know what it means and they won't be able to tell us about it. So, yeah.
Darn.
So then you can also hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a subject you would like us to cover or address, a suggestion for an interview, a haiku that comes to the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.